Hi, physiology students. Welcome to your first lab. This is on homeostasis and negative feedback systems. And you'll see this is the first lab listed in your lab manual, which I'll show you in a minute. These intro videos, I'll try to keep pretty short just to encourage you to watch them all the way through. I'd also encourage you to email me if you have any questions. I'll also set up a discussion post for Lab 1 where you can discuss any questions that you might have with other classmates. So homeostasis, we'll talk about this in lecture, is a relatively constant internal environment, and it's maintained by cells, tissues, and organs that operate through what we call negative feedback systems. And a negative feedback system, basically if any sort of variable like body temperature, blood sugar, glucose levels fluctuates around a set point, which is the normal or average accepted value of that body temperature or glucose level in the body, external factors will stress the system and cause the variable to increase or decrease. And the physiology behind that is what we're going to go over. Negative feedback will detect and change and move that variable back to its set point in the organism in order to keep the organism alive. So any sort of disturbance will send an input to sensors in the system, which will take that measuring element and determine how it relates to the set point. If there is an error, if it's too high or too low from that set point, it'll send a signal to the controller, which is usually a place in your brain or thalamus. And that command sender will send a signal to any sort of effector, whether that's an organ, a muscle, a tissue, a cell, to try to change that disturbance back to a normal set point value. And that's called negative feedback. It reverses the change, tries to get things back to normal, which is the set point in the organism. Controlled variable and set point. So the controlled variable will be the variable that we are detecting, whether that's body temperature, glucose levels, heart rate. And this is a graph of a controlled variable, shows the variable fluctuating around a set point. And a normal range of values is a range of measurements that includes most healthy people. So not everyone has the same heart rate or blood glucose level or exact same body temperature at all times. So there's a normal range of values that includes healthy people. And then the controlled variable should be within that normal range of values. And again, the set point is usually the average of the normal range of values in one healthy individual. Homeostasis is constancy of the internal environment. The next few slides are actually taken directly from the Chapter 1 lecture, which hopefully you've seen by now. The main purpose of our physiological mechanisms is to maintain this homeostasis or balance. Deviation indicates disease, and homeostasis is accomplished usually through negative feedback loops, which is what we just went over. The pathway is what you should be familiar with, so reference these slides as you're filling out questions to your lab report later. Sensors in the body will detect changes, and these sensors can be in the skin, they can be in muscles, and they will send any sort of change to the integrating center, which assesses that change around the set point. The integrating center is usually the thalamus in the brain, and it'll send those instructions to an effector, which can make the appropriate adjustments to counter the change from that set point. And again, the effector can be blood vessels, it can be muscles. Then the mechanism continues. It will move in the opposite direction from the change in a negative feedback system. It'll make the change from the set point smaller. It will reverse the change in the set point. This is a continuous process. Your body's always making fine adjustments to try to maintain homeostasis. Again, whether that's body temperature, this is why right now, if it's 100 degrees outside, you're probably sweating. If you're cold, your body will automatically initiate shivering to try to warm up. So this is the negative feedback loop, taking into account what a sensor is and what an effector is. So here are, let's say, the normal range of values. This would be on the y-axis of the graph, and the x-axis shows time. So if there's a change in the set point, which is signaled by the rise in this graph, the sensor will become activated. So it'll sense that change, send it to the integrating center, which is signified by this plateau in the graph, and then it'll tell the effector what to do to try to bring this set point back to normal. And that's why the graph is going back down to its normal set point. The normal range, this is where the set point is up here. The sensor will be activated if the set point decreases, send it to the integrating center in the brain, and tell the effector to bring that set point back up to normal. Body temperature is an example of a negative feedback loop. Sensors in the brain will detect any sort of deviation from 37 degrees Celsius, which is 98.6 degrees Fahrenheit. Another part of the brain will assess this as actionable, meaning we have to do something to change this. And effectors, your sweat glands, are stimulated to cool the body. Once the body is cool, sensors will then alert the integrating center in the brain, and the sweat glands will be inhibited or stopped because they don't need to be working anymore. And the end result regulates the entire process. Production of the end product shuts off or downregulates the process, and this is why it's called a negative feedback loop. Antagonistic effectors, homeostasis is maintained by opposing effectors that move in opposite directions. And the best example of this is when you're hot, you sweat, and when you're cold, you shiver. And this is just an example of antagonistic reactions. And again, other examples, blood glucose levels, blood calcium levels, which have to do with your skeletal system, your heart rate, and your blood pH. And again, this is just a great graph showing the set point or average of a normal range of values in a person, of whether it could be body temperature, blood glucose levels. And the arrows, the pink arrows going up and down, are signifying the change of that set point. So you can see how the negative sign within the blue circle shows that a negative feedback system has been activated to try to bring that value back to its normal set point. And the bottom graph is showing normal body temperature is 37 degrees Celsius. That's the set point. And if that decreases, you'll start shivering to try to increase your body temperature. And if you get too hot, you'll start to sweat. So this is a great way to kind of depict, and you can use graphs like this in your lab report procedure questions as you're trying to depict what happens as ranges of value change around a set point. And when the systems are activated to try to change that value back to normal. Quantitative measurements, these are just, scientists must measure specific values and then determine the range, the averages, and their deviation from their averages. And again, the average is the set point, and the normal range would be the normal range of all values in a healthy individual. And this is important for diagnosing disease, but also assessing effects of drugs and other treatments and experiments. So this is just a table of examples of normal range of values in your blood pH, bicarbonate levels, and it goes through urea levels, amino acids, protein, glucose levels is a big one if you're a diabetic. So these are just normal range of values for someone, for measurements of some fasting. So that's pretty normal to be fasting when you take a blood sample. The regulation processes within organs can occur intrinsically, where cells change in signal to respond, or extrinsically, where the brain or other organs regulates an organ using the endocrine system or your autonomic nervous system, being the parasympathetic or sympathetic divisions. The nervous system innervates organs, and the endocrine system releases hormones into the blood, which transports them to multiple target organs. And we'll learn a lot about the endocrine system and how that will regulate these set point values using negative feedback mechanisms. So this is a look at neural, so nervous system, and endocrine, which is hormone regulation. If you're lying down and all of a sudden you stand up, you'll notice that your blood will rush from your head and it'll pool at your ankles. And this will cause your blood pressure to fall. You might feel a little lightheaded. So that blood pressure falling will be a stimulus that your body perceives. The blood pressure receptors will respond as the sensors, and it'll send that sense through sensory nerve fibers to the brain, specifically the medulla oblongata of the brain. And we'll learn some of these pathways. Then it will send out a motor response to your heart rate, which will tell your heart to start pumping faster to increase the heart rate and to try to increase your blood pressure back to normal to try to get blood flowing back to your brain so that you're not lightheaded anymore. Feedback control, hormones are secreted in response to stimuli. Secretion can be inhibited or inhibited. Let's see, I'm going to go through, this is a good example of blood glucose levels, whether you're eating or fasting, and how blood glucose levels change through negative feedback mechanisms. This is showing temperature, just another way, whoops, sorry about that. This is just a flow chart. So I'm just going to take you guys, I'm going to share a different screen here of your lab manual itself. So this, you should see your lab manual on your screen. So you can read through the homeostasis and negative feedback systems introduction, and then we'll go through the procedure and you'll use the outline on the following page to construct a flow diagram of body temperature. And this takes us through, you'll also talk about resting heart rate. So the first procedure, you'll want to fill out this flow chart, and you don't necessarily have to send this in to me. You can draw it out and send in a picture if you'd like. But in this flow chart in body temperature, we'll kind of go through it together right now. So if we start here right in the middle, if the set point, basically if your body temperature decreases below -37 degrees Celsius, that'll be sensed by the cells in your skin, and it'll tell your integrating center or your brain to tell effector organs, which would be, in this case, muscles, but also it'll tell your blood vessels to dilate. So if you're extremely cold, you'll have sensors in your skin which pick up on that. Your integumentary system, your skin is your first line of sense response, and it'll tell your brain to send out kind of a motor response to your muscles to start shivering. And shivering will hopefully help to increase your body temperature back to normal. And vasodilation, which is your blood vessels dilating or getting larger, will help to dissipate the heat, which will help to cool your body. Then if your body temperature increases above normal, and I'm so sorry, I told you vasodilation, if your body temperature decreases, your blood vessels will constrict to try to keep the blood in the center of the body to increase temperature. So if we go in the opposite side of the flow chart, if your body temperature increases, that'll also be detected by sensors in your skin, which send that to your brain, and your brain will send this out again to your skin to start sweating, to try to sweat or release some of that heat, but also to your blood vessels to vasodilate to try to dissipate some of that heat. So the procedure, you should do this first procedure on your own. You're going to take your heart rate in beats per 15 seconds and then beats per minute. So you'll place your index and middle finger over your radial artery in your wrist. You'll count the number of beats to 15 seconds and place that in this number 1 spot. And then you'll multiply by 4 to get your heart rate down here. And you'll do this 10 times in a row. You'll calculate your average, which will be your set point. We won't be doing this in a group, but then you'll calculate your average. You'll calculate out your pulse rate in beats per minute in each of the 10 trials that you did. And then you'll calculate out the average. So you'll add them all together, divide by 10, and you'll use a dashed line across the graph to indicate your set point. You'll also tell me your range of values, which will just be the lowest heart rate to the highest heart rate. So that's how you will do the first procedure, the pulse rate graph. You can skip the class averages for now, and then in that discussion post that I start for lab 1, you can ask people, hey, my heart rate was this, is this normal? Then you'll answer these lab report questions. You'll use your flow diagram of body temperature to predict what changes and responses will happen to jumping into a cold swimming pool. And then you'll graph the changes in temperature and the effector responses. And then you'll indicate on the graph the point when the effectors become active. And for this one, you can kind of go back to my PowerPoint and use some of these flow charts and graph that I gave you an example of. Specifically, this one is a great graph just showing what happens when the set point, when your body dips below 37 degrees, which is happening if you jump into a pool, your body will begin to shiver and your blood vessels will vasoconstrict, so they'll try to keep all the blood close to the heart. So I'm just going to go back and share the lab manual again. Produce a flow diagram of resting heart rate regulation. So what keeps your heart rate in normal resting rhythm? Which part of the autonomic nervous system helps maintain homeostasis? You might need to use your textbook or other resources to answer some of these questions as well. Every time you visit the doctor, someone will take your temperature, pulse rate, and blood pressure, and why. And if your resting pulse rate were too high, what would the doctor do? And how is homeostasis important for cell survival? For example, what can happen to cells if body temperature increases above normal? So again, that takes us through the end of lab 1. So really you'll want to, you can write out these questions on a sheet of paper and take a picture and send it to me that way, because you will be doing some flow diagrams and graphs, because I'll want to see, specifically I want to see your graph for the pulse rate graph as well. Or you can type this all up in a Word document and upload a file that way to Canvas. If you have any questions about this, please let me know. I'll start a discussion post and try to be in that often this week to help you answer some questions there. Just keep in mind that each lab is due every week on Sunday at midnight. So the labs will sneak up on you because you will have one every week. Hope you guys are all doing well. Take care.